Hi, welcome to my final video for CIS 3620. Decided to do my final video on healthcare and agricultural science and how they'll be affected by CRISPR. Alright, so what is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. So that doesn't really mean much to us, but in layman's term, it means pretty much that there's a bacterial gene repeat that's spaced between copies of viral DNA. And that viral DNA is added into the bacteria as sort of a checklist to know things to destroy in its system. So with CRISPR in healthcare, scientists have found a way to extract the Cas9 protein from these bacteria that act like a pair of scissors that can cut the DNA any way they need to. So this could essentially be used to edit out any bad gene and add in a new good gene pretty much anywhere, and the process could easily treat virtually any disease. For example, immune cells could be taken from someone's body and create proteins that can attack cancer cells, and in embryos this could be used to edit out common known diseases before anyone's even born, and this could also possibly replace antibiotics. So with CRISPR in agriculture, essentially CRISPR can be used across any species that they've come across so far, and it's already being used on crops, which can allow for more resistance against pests and reduce the amount of crops that are lost to disease. For example, some Chinese scientists have created a strain of wheat that is resistant to powdery mildew that's a really destructive fungal disease. So one of the most impactful effects of CRISPR could be the gene drop, which this is a process where they can actually encode CRISPR into the genome to allow for self-editing. And this self-editing could be sent through the line to eventually where a whole entire population of an organism could have this gene drive effect. And essentially this could be used to eradicate entire diseases, from things such as malaria from mosquitoes and Lyme disease from ticks. So all that being said, there are definitely dangers to the CRISPR system. The question is, where's the line drawn? Because within 20 years this type of editing will virtually be mastered, so it's hard to say what we should and shouldn't do with this. Human embryos can be edited in any way, shape, or form, and it's kind of an ethics thing as to, you know, they don't have a say in what's being done to them because there's no way to, and it's just a big question to be drawn. And as for the gene drive, cascading effects could be completely catastrophic and unforeseen. With eradicating something as malaria, there's no way to tell the effects of what could happen from that. All right, and that's my video. Thanks for watching.